This is lecture 2 on chapter 5, so it is chapter 5-2 on neurotransmitters. In the first part we'd been talking about synapses. It was the last part we had discussed that there are both excitatory and inhibitory synapses. Excitatory synapses will depolarize the cell membrane. Usually we'll find this on the dendrites, and the inhibitory, as we said, were usually found on the soma. So the inhibitory synapses tend to be sequestered on the soma, axosomatic types or proximal dendrites, but there's also a smaller number or density of inhibitory synapses on the dendrites all the way out to the tip. Especially here when we see these axospinous types of synapses, typically there would be inhibitory synapses on the neck of the spine and excitatory on the head of the spine. So there's a lot of integration that can go on here between just affecting the postsynaptic membrane polarity or millivoltage, such that you integrate here and can either make it more challenging for the excitation to reach the main part of the dendrite and become a graded potential heading for the spike zone at the axon hillock to contribute to the generation of an action potential or a firing rate or a firing pattern, or you can make it more difficult by hyperpolarizing with these inhibitory inputs. So there's integration there on a small scale, and on a grand scale the same thing happens at the soma, and hyperpolarize the membrane here, and then graded potentials can be diminished or canceled out even, coming from the entire dendritic tree or just a portion of dendrites and so on and so forth. So we had discussed that a bit, and we'll continue to work on this idea as we proceed. In this illustration we are considering the effects of stimulating afferents, electrical stimulation of just one in a subthreshold fashion, two stimuli or two stimuli closer together to demonstrate temporal summation, or two separate inputs to demonstrate spatial summation. In this case we're looking at an excitatory input and an inhibitory input onto the soma. So we see with this time separation, and of course time is on the x-axis and membrane potential in millivolts is on the y-axis, resting membrane potential at minus 75, threshold at minus 55. So if these two subthreshold events, synaptic events, are separated in time sufficiently so that the first one has decayed adequately such that the second does not thereby reach threshold, there is no action potential fired. The temporal summation, if they are these two stimuli and the same afferent are close enough together in time, causes the summation of the membrane depolarization, opening enough sodium channels when the graded potentials reach the axon hillock to generate an action potential that will of course be conducted down the axonal membrane in the typical all or none fashion. Spatial summation, of course, is when the two are separated, and in this case, I'm sorry, these two are excitatory. This is an example of an inhibitory and an excitatory. So if these two are stimulated at the same time, both E1 and E2, the dual excitations, when their graded potentials reach the axon hillock, threshold is reached, enough sodium voltage-gated sodium channels open, and you cause an action potential. Now remember, these are all cases where the membrane potential is that resting membrane potential. If we have metabotropic receptors, say some neuromodulators coming in and opening calcium channels or inhibiting potassium channels, then the membrane will depolarize and we will not be at resting membrane potential. We might be closer to threshold, so any of these events will more readily cause an action potential or sustain firing at a certain rate. Here in this example we're looking at spatial summation, actually a cancellation almost, of an postsynaptic inhibitory postsynaptic potential, or IPSP, mediated most likely via GABA, opening a chloride channel with chloride influx, making it membrane more negative on the inside. You hyperpolarize, and then here you excite very likely sodium influx, and they're both the excitatory and the inhibitory are stimulated together here at this second stimulus event. So the hyperpolarization and the depolarization, similar to what we would have seen over here, are summated in a subthreshold event. If you had a neuromodulator come in and at this point would hyperpolarize the membrane and keep it there, if you then stimulated just the excitatory drive, you'd see a similar type of an effect. And again, we're recording close to the axon helix so that we can get the graded potentials, summated or unsummated, recorded, as well as the action potential if it is generated. Now we transition over to neurotransmitter biosynthesis and storage, and we note that precursor molecules are produced on the ribosomes of the ER and the nerve cell soma and transported in vesicles to the axon terminals. Synthesis is completed at the synaptic vesicles. Immediately in your mind, this should tell you that this is describing the synthesis of peptide neurotransmitters, not the neurotransmitters that depend upon biosynthetic enzyme molecules. Therefore, we would have to rephrase this. The biosynthetic enzyme molecules are produced on the ribosomes, so their genes are expressed. The mRNA is either translated in the free ribosomes, as we'll see, or the fixed ribosomes on the ER. And the biosynthetic enzymes will either be sent directly to the cytoplasm or into vesicles after the ER and Golgi, and immediately upon generating vesicles from the Golgi, we have the capability between the cytoplasmic and the vesicular biosynthetic enzymes for synthesis of the classical neurotransmitters that we've described in the first part of this lecture, other than the peptides. And then the vesicles synthesizing and storing the transmitters will be transported to the axon nerve terminals, and as well, synthesis continues at the synaptic vesicles in the synaptic terminals. So that distinction, of course, can be shown by this illustration, focusing just now on the neurotransmitters other than peptides. So the genes for the biosynthetic enzymes are expressed. The RNA goes either to ribosomes on the ER if the biosynthetic enzyme is to end up inside the vesicle, or on free ribosomes if the biosynthetic enzymes are to end up in the cytoplasm or axoplasm. They all get transported somehow with just the bulk flow of axoplasm or with the anterograde vesicular transport, so that immediately upon generating the synaptic vesicle after the Golgi, you've got the synthetic enzymes in the cytoplasm of the soma and within the vesicles, and everything else is available, so you can begin to synthesize the neurotransmitters in the soma. It even gets into the dendrites. When you use methods to stain for the transmitters, you find them in the dendrites and the soma, as well as along the axon and in the terminal. When we start looking at peptides, you'll see that differs depending upon the type of antibody you use to detect the pre-prohormone or the mature biologically active molecule. So the synthesis ends up filling the vesicles with the neurotransmitters. There is a ready releasable pool, the storage pool as we've described, and that at the presynaptic grid for release. And of course the influx of calcium at the presynaptic membrane causes the exocytosis here. Calcium, voltage-gated calcium channels are also located along the pre-terminal portion here and interact with the cytoskeletal components to bring the vesicles close to the presynaptic grid for this calcium to trigger the exocytosis. So calcium from here takes it from the readily releasable pool, the storage pool, to put vesicles close so that they can be released. This slide is showing reuptake through endocytosis. This has been demonstrated to occur with some peptides, but is not the mechanism for uptake and removal of the classical neurotransmitters from the synapse. But this does occur in all neurons. There is a retrograde transport with endocytotic membrane recycling, even regenerating of vesicles that can be recharged with newly synthesized molecules. So there's a bit of additional information on this slide that you need to make sense of, and you should be able to integrate it into your understanding of neuropeptides versus the neurotransmitters, here distinguishing them based upon the mode of synthesis. Continuing with the synthesis, of course, glutamate and aspartate are amino acids. They can be synthesized from glucose, so the general metabolic pathway is a source of glutamate and aspartate, but it's thought that there's separate membrane transport proteins to get these into the neurons and target the vesicles for release through exocytosis. Acetylcholine is another one we talked about. It's made through the synthesis of the biosynthetic pathway by which choline acetyltransferase acts on choline such that acetyl-CoA is transferred onto the choline molecule. And this is the synthetic enzyme that you need to know and recognize, and its acronym CAT, choline acetyltransferase, for the synthesis of acetylcholine. And I have an asterisk here as if that goes there, but let's look more closely and see that indeed this is wrong. I don't know what I was thinking at the time. I didn't look at the structure of the molecule. This is glutamic acid, the amino terminus and the carboxy terminus. It has an additional carboxyl group on it. So we go from that to this structure that is decarboxylated. So there must be a decarboxylase enzyme here, not this one. If indeed this was CAT, we'd have to see a choline molecule and an acetyl-CoA molecule. So we know this is an error on labeling the slide. So what enzyme would be here? Well, this is glutamic acid. So glutamic acid decarboxylase is the enzyme for the synthesis of GABA, gamma-aminobutyric acid. Alpha, beta, gamma amino, 1, 2, 3, 4 carbons, butyric carboxylic acid. So this is GABA synthesis here, and the synthetic enzyme is GAD or GAD, glutamic acid decarboxylase. Now we can consider the synthesis of the monoamines, the catecholamines, and the indoamines. You'll notice that I have corrected the spelling of the indoamines. In this case, we're talking about serotonin. Another slide mislabeled. There should be another major heading here. Indoamines for serotonin. And the catecholamines, of course, include dopamine, norepinephrine, and epinephrine. And we see the precursor is an amino acid, tyrosine, that is enzymatically converted to dopa. This is such a common acronym for this now. When I tried to look it up in a few sources on the internet, they're just showing dopa and not the full chemical name. But I'll leave it to you to try to find out what this acronym stands for. Enzyme that acts on that is dopa decarboxylase. We'll see that it's the same one that is involved in the synthesis of serotonin, the final step, and I will call these they are identical enzymes, identical genes in two different populations of neurons. It is aromatic amino acid decarboxylase. AADC is the term I will use for it. So in dopamine neurons, it's called dopa decarboxylase. In serotonin neurons, it's called 5-HTP decarboxylase, but it is exactly the same enzyme in both neurons, populations of neurons, and the same gene product. Continuing then, dopamine is produced, enzymatically converted to norepinephrine, enzymatically converted to epinephrine, and we'll look at this in detail in just a moment. As well, we'll look at the biosynthesis of serotonin from tryptophan. misspelled. Oh, goodness gracious. Tryptophan hydroxylase is the enzyme here. And the one here is tyrosine hydroxylase. They are both in the general category of mono-oxidases, amino acid monooxidases. So there's tyrosine hydroxylase converting tyrosine to dopa, and tryptophan hydroxylase converting tryptophan to 5-hydroxytryptophan. And then the decarboxylase converts it to 5-hydroxytryptamine serotonin. And we'll look at those biosynthetic pathways in the next couple of slides. First, we're looking at this figure from our textbook where tyrosine is the precursor, an amino acid. Of course, exits the plasma from down its concentration gradient into the interstitial fluid compartment. As I said, it is thought there are two sets of amino acid transporters, those that are dedicated for uptake of precursors. Remember the amine precursor uptake and decarboxylase system, the APUD system, common embryological development. Well, it's thought that the uptake for the precursor is a separate one from just the general uptake of tyrosine as an amino acid to be used in protein synthesis in general. Anyway, we get the tyrosine uptake anywhere along the neuron, soma, dendrites, and the axon as shown here. We're just focusing on synthesis at the terminal. And the tyrosine is then within the cytoplasm or axoplasm, cytoplasm of the axon. Tyrosine hydroxylase is the first enzyme. We notice the negative feedback of some of the end products, the dopamine and norepinephrine. This is Na for noradrenaline. We convert it immediately in our minds to norepinephrine, the accepted terminology. So they both have negative feedback on this. Then this is the rate-limiting enzyme. The tyrosine, we'll look at the chemical structures of these molecules and the cofactors for the enzymes in just a moment. Dopa is produced in the cytoplasm, dopa decarboxylase, AAA-DC, aromatic amino acid decarboxylase, remember, identical for catecholamines and for the indoamines, serotonin. The dopa decarboxylase converts dopa to dopamine. And then there is a vesicular monoamine transport protein, VMAT, vesicular monoamine transport protein, that will transport dopamine into the vesicle, as well as it will act on norepinephrine when it is taken back up from the synapse to recharge vesicles. So that's why it's vesicular monoamine. It will act on all the monoamines. And the norepinephrine is then produced by dopamine beta hydroxylase acting on the dopamine in the vesicle. The noradrenaline is stored within the vesicle. And if there is another gene expressed in this neuron for the synthesis of epinephrine, then in this vesicle there will be another enzyme, phenylethanolamine N-methyltransferase, or PNMT is the acronym, dopamine beta hydroxylase, DBH, tyrosine hydroxylase, TH, and then AAA-DC for the dopa decarboxylase, as we had stated. And then adrenaline would be released from this neuron through exocytosis, the vesicular release. So think of the different catecholamine neuron populations. Dopamine, the enzyme gene expression would stop at AAA-DC, but the dopamine, once synthesized in the cytoplasm, would be transported into the vesicles to charge the vesicles for release. Previous slide showed us the two populations of vesicles, those that are stored basically, and then the other pool at the presynaptic grid, so keep that in mind. If a neuron is going to be norepinephrinergic or noradinergic, then it has to express 3 genes all the way up to the dopamine beta hydroxylase. If it is going to be adinergic, it must express the genes all the way up to the PNMT. So keep this in mind. Differential gene expression at the level of the cell, cell-specific gene expression. At the tissue level, all 4 of these genes for these gene products would be expressed in the adrenal medulla because adrenaline or epinephrine is the primary molecule of release. Here then we can look at the chemical structures, the enzymes, and their cofactors here in this noradrenergic cell that is only going to express the genes for the first three enzymes in this series, such that norepinephrine would be the final molecule released. So we see tyrosine, the amino terminal, carboxy terminal, basically the same, and there is this phenolic ring or catechol ring, therefore catecholamines. Tyrosine hydroxylase is going to add a hydroxyl group onto this carbon here. This is carbon 1, 2, 3, 4, 5, 6 of the phenol ring. So in the 5 carbon position of the phenol ring, TH adds a hydroxyl group. Reduced pteridine, oxygen, and iron are the cofactors. You will have to know these cofactors. You'll have to be able to spell them and type them in for the exam on short questions. You'll have to recognize each of these structures as the corresponding molecules. Not so much DOPA, but anyway, the AAADC decarboxylase will decarboxylate at the first carbon, the alpha carbon here. Remember, this is alpha carbon, this is beta carbon. Very important when we get to dopamine and convert to norepinephrine. So dopamine has the 2 hydroxyl groups on the 4 and 5 carbon position, and there is another hydrogen on the alpha carbon. The carboxyl group is gone. Pyridoxal phosphate is a cofactor for the AAADC. The dopamine is converted to norepinephrine by dopamine beta hydroxylase. Ascorbic acid and oxygen are cofactors. So alpha beta hydroxylase adds a hydroxyl group to the beta carbon in this molecule. So a hydrogen is removed and replaced with a hydroxyl group to produce norepinephrine. I give you the link to the website where I found this illustration. It's very nice. When you click on these various boxes, it takes you to other links and other information about the enzymes. So the biochemistry of the nomenclature of the enzymes is highly standardized, and I encourage you to familiarize yourself with it again. So here's L-tyrosine. There are alternate metabolic pathways, but the tyrosine hydroxylase with the tetrahydrobiopterin and oxygen as the cofactors mentioned in addition to the others, and that is converted to 4-alpha hydroxy tetrahydrobiopterin to produce L-dopa. Again, we add the hydroxyl group on the 5 carbon, and this dopa can be converted to melanin through melanin biosynthetic pathways. So if it is not readily converted by the aromatic amino acid decarboxylase, the AAADC, then it will be shunted in the other direction. And melanin does accumulate in catecholaminergic neurons, especially dopamine neurons. Here we see the dopamine, so an alternate structure that you should be able to recognize in addition to the previous slide. And the dopamine beta hydroxylase is shown here with the ascorbate and oxygen being converted to dihydroascorbate and water as the byproducts. And the next intermediate in the synthesis of adrenaline is noradrenaline. And this is the dopamine beta hydroxylase. I'm sorry if I called that the wrong enzyme. AAADC is here. This is the dopamine beta hydroxylase. So we have hydroxylated the beta carbon shown there. So this is the norepinephrine or noradrenaline. Then the final enzyme, the PNMT, phenylethanolamine N-methyltransferase. So this molecule is also called phenylethanolamine. We have the ethanol here, phenyl, and it is 2 carbons. So ethanol, amine, phenylethanol, amine, N-methyltransferase. Phenylethanolamine N-methyltransferase, we're going to transfer a methyl group onto the nitrogen here, and we see that the cofactor here, they're not spelling it out, but it is S-adenosyl L-methionine, or SAM. You may have remember that abbreviation. Common methyl group source or provider, and plus the phenylethanolamine leads to S-adenosyl L-homocysteine plus the N-methylphenylethanolamine. So we N-methylated it, so N-methyl phenylethanolamine, or adrenaline norepinephrine. A lot for you to remember, but work on it, because I will test you on each of these. Next, then, the indoamine synthesis. Notice that there is an E in indoamine. Tryptophan is the precursor, so it not only has the 6-member carbon ring, but the 5-member ring here, of course, a nitrogen in it, and as well, it is an amino acid with the amino and carboxy groups on it. Tryptophan hydroxylase is the first enzyme to convert tryptophan to 5-hydroxytryptophan, or 5-HTP, and 1, 2, 3, 4, 5. So we add a hydroxyl group to the 5-carbon on the phenolic ring. We have the tetrahydrobiopterin and oxygen and iron as the cofactors. Remember, these are monooxygenases. The hydroxylases, amino acid hydroxylases, are monooxygen aces, so it uses one of these oxygens in this process. The other oxygen ends up with 2 of the hydrogens from the tetrahydrobiopterin to form water, and we end up with, of course, the alpha dihydrobiopterin. And of course, the iron is contributing, as iron does, for the redox reaction. So the 5-hydroxytryptophan is converted by the AAADC, the aromatic amino acid decarboxylase, AAADC, or 5-HTP decarboxylase in the serotonergic neurons, to decarboxylate, and we end up with 5-hydroxytryptamine, 5-HT, or serotonin. The serotonin, as we said, is involved in the synthesis of the other indoamine, melatonin. These are bidirectional. All enzymatic reactions, as you know, are bidirectional. So if 5-HT N-acetyltransferase, or NAT, in the pineal gland acts on serotonin, then it will produce the intermediary structure, the N-acetylserotonin. Excuse me. So there's an acetyl group added here. There's the N, and then here's the acetyl group. And then 5-hydroxyindole O-methyltransferase, or HIOMT, will then convert N-acetylserotonin to melatonin. So the 5-hydroxyindole O-methyltransferase will methylate at the hydroxyl group on the 5-carbon of the phenolic ring. Hope you can remember all that. Okay, receptors then, we introduced them now, and we'll go into detail in later lectures on other chapters, from other chapters. So they can either be ionotropic or metabotropic. The ionotropic can also be 2 categories of ion channels, either voltage-gated ionotropic or ligand-gated. So now that we're talking about receptors for neurotransmitters, we're talking about ligand-gated ionotropic receptors. The alternate are the metabotropic receptors, and 2 pathways that we will focus on are the adenylate cyclase pathway for production of cyclic AMP and the IP3 pathway, and we'll detail those when we get into receptor mechanisms. We just want to introduce them now through these next couple of slides. So we recognize the axon terminal, the action potential coming down, our vesicles arriving, The synaptic vesicles contain the neurotransmitter molecules, as we know. Here we can see some of the calcium influx in the pre-terminal portion to interact with the cytoskeleton, move the vesicles toward the presynaptic grid. The exocytotic event, once the action potential reaches this, and you get the local increase in calcium here through the voltage-gated ion channels, you get the exocytotic event, contributing to the synaptic concentration of the neurotransmitter that diffuses across to target either an ion, a ligand-gated ion channel, or metabotropic receptor that can affect nearby ion channels that might be voltage sensitive, or if they're altered, typically phosphorylated or dephosphorylated, then they'll open or close or be more sensitive to voltage changes. All these types of parameters can alter these types of non-ligand dependent ion channels in the nearby membrane, not just within the synapse, but all over the membrane. So you change an enzymatic event in here. Cyclic AMP is diffusing around, activating phosphokinase, protein kinase A, the A for cyclic AMP, that can affect these. And we'll see that when we proceed to the next slide. So the neurotransmitter would bind and ligate the ligand-gated ion channel. The ion opens through conformational change. In this case, it is a sodium-dependent channel. This could be glutamate on NMDA and N-methyl D-aspartate channel. Excuse me, it would be non-NMDA channel. The NMDA receptors are both voltage-gated and ligand-gated, but let's not get into that now. This would be a non-NMDA or AMPA type of glutamate receptor. I got you going now. Anyway, ligand gate, conformational change, open, sodium influx, more positive ions on the inside of the membrane. You increase the membrane potential, depolarize it, and progress toward threshold, as we saw. Degrade it, or it just comes off. It's a dynamic event. This isn't a permanent binding. It's on and off, on and off, on and off. The more frequently it is ligated, the more frequently the channel is open. If the synaptic concentration is not sufficient to keep it ligated on an ongoing basis, then the ion channel will close. Now, they're showing destruction of the molecule, but there's several different means of removal from the synapse, as we will see. Here we're looking at a water-soluble extracellular chemical messenger as well that depends upon an integral membrane protein receptor. When it is ligated, there's a conformational change mediated through G proteins. We'll look at a little bit of detail in a moment. And activate the enzyme. In this case, adenylate cyclase, ATP, will be converted to cyclic AMP, activates protein kinase, in this case protein kinase A. Protein kinase A is now active, and it can activate other enzymes or act on ion channels and other receptors, and so on and so forth, cascading to affect maybe a rate-limiting reaction such that you get the physiological effects of the hormone. As well, we know that the protein kinase A phosphorylates CREB, cyclic AMP response element binding protein. When phosphorylated, can bind to genes and affect gene transcription. So one second messenger can affect many different aspects of the target cell, all of which are targets in a neuron, including membrane ion channels and other receptors possibly. So here is the ATP, and this is the cyclic AMP. Could you recognize that? So here we're looking at the voltage gated ion channel and a ligand gated ion channel. In this case, it's the nicotinic cholinergic ligand gated, so two acetylcholine molecules have to bind to it, and that causes the conformational change, and the channel would be open. I don't know if they're intending this to be the unligated form, but we could consider it unligated in our imagination. Once it's ligated, it's open. Or if it is voltage sensitive, then this sodium influx via the nicotinic acetylcholinergic receptor will change the polarity of the membrane nearby and possibly open it. If we have a protein kinase A here or some other molecule that can affect this, maybe phosphorylate it, make it either easier or more difficult to open it, and impact this nearby ion channel. We can see the illustration of the beta adrenergic receptor for norepinephrine and epinephrine. You may know that it's has a higher affinity for epinephrine, but anyway, a beta agonist like epinephrine would bind to the integral protein and cause a conformational change. In this case, it is a G sub S G protein complex. There's an alpha, beta, and gamma component subunits to this, so it's a complex of proteins. GTP will bind to it in the ligated form, and then the alpha subunit can diffuse away, and in the membrane, as long as it has a GTP molecule on it, and encounters adenylate cyclase, and then it will convert the ATP to cyclic AMP. The protein kinase A is in a complex. There's the regulatory element and the catalytic element. The cyclic AMP acts on it to cause a cleavage. So the catalytic subunit then can phosphorylate a nearby calcium channel. And of course, the alpha subunit can also directly affect that calcium channel. So we see a couple of different mechanisms by which a metabotropic receptor, G-coupled, can affect a nearby ion channel, in this example a calcium channel. So we can follow through this sequence in a more globular type of illustration, simplified here below, where it's not showing the three subunits of the G protein just as one. But I want to make another point as we're going along. In the unligated form, the receptor, the binding protein, it has a higher affinity for the G protein complex, or throughout the G protein complex for the alpha subunit, because GDP is actually bound to it, as you can see. When it becomes ligated, then the GDP falls off, GTP jumps on, and the affinity for the alpha subunit decreases, and the alpha subunit with the GTP on it now can move through the membrane, and in this form, the alpha subunit that is with the GTP on it is the ligand for the high affinity site on the adenylate cyclase, so it will now stick to that and let go of the original complex. So it's changing affinities that are very important to understand the concept I'm trying to emphasize here. And again, activate the adenylate cyclase, and you get the dipyrophosphate and the cyclic AMP activating enzymes that can target gene expression and impact protein synthesis at the nucleus, and have a divergent pathway for changes in membrane permeability and potential, possibly targeting potassium and calcium ion channels, as we have discussed throughout. So if we think of these not as stimuli, but as metabotropic events where we can get changes within the neuron, allowing either potassium to stay in the cell, decreasing potassium efflux, or increasing calcium influx, or maybe increasing sodium influx, then varying degrees of activation of the adenylate cyclase or a second messenger system cascade within the target cell can bring the membrane potential up to different levels, and thereby affect the firing rate of the target neuron. And this firing rate might be spontaneous, or it may be driven by other excitatory inputs. So we want to keep that complexity in mind. As you recall, we've talked about these type of phenomena for neuropeptides. Here, for example, the substance P increasing the membrane potential such that when we electrically stimulate, we get a pulse of action potentials. and the interaction of somatostatin and acetylcholine. Here, the somatostatin through metabotropic mechanisms altering the response to the nicotinic cholinergic receptor that alone causes only these increases in firing. So this is transmitter-driven firing rate, an excitatory transmitter-driven firing rate in this neuron, whereas this is the spontaneous activity of the neuron, where the somatostatin decreases the spontaneous activity but enhances the transmitter-driven activity. So you see that's a different type of neuronal integration. Stop paying attention to yourself and begin paying attention to your synaptic input. So, and we also have seen this illustration of the action potential. We just want to remind ourselves that we're considering the membrane potential. If it's resting, it's at -70. It may be anywhere above or below that, especially mediated through metabotropic events. It will have a flux up and down depending upon ionotropic events that are either depolarizing or hyperpolarizing. It's all integrated on the entire neuron, but at the beginning of the axon hillock, where the spike zone is, it'll determine if enough voltage-gated sodium channels that are there and probably the highest density of anywhere in the neuron, other than the axon itself, right? So if enough of those voltage-gated ion channels open at the axon hillock, an action potential is going to occur, with the sodium influx causing the rapid rising phase. They are closing, closed, they're opening, opened, closing, closed, and we'll discuss a little bit more detail this absolute refractory period based upon the nature of the voltage-gated sodium ion channels. And then the potassium channels, voltage-gated potassium channels finally open, even though they begin opening at threshold. They open slower, so they contribute to the rapid repolarizing phase and the hyperpolarization. This can be altered, and this can be altered, and there isn't much that we can do here through metabotropic mechanisms, at least. So what we're going to focus on is the relative refractory period and how we can change that and how it can affect the maximum firing rate of a neuron. So first, let's think of the voltage-gated sodium channel starting here at number 1. It is closed because the membrane is at resting membrane potential here. So there's no sodium influx, no potassium influx, although I think they're showing permeability. But as permeability increases, there'd be potassium efflux, and sodium influx. As the permeability of sodium increases, there's going to be sodium influx. But no potassium channels are open yet, even though they start their conformational change at the same time. The voltage-gated sodium channels are more rapid. Because we're looking at an action potential, we're thinking of the axon hillock, where it has a very high density of these two, as well as everything else that's responsible for resting membrane potential, such as the sodium-potassium ATPase. Anyway, the timed event. Voltage-gated sodium channels are voltage sensitive for opening, voltage dependent for opening, and time dependent for closing and being reactivated. So let's follow the sodium first. Fully opened after it has membrane has depolarized sufficiently at threshold, so a large enough population of voltage-gated sodium channels open, and you get a dramatic increase in permeability, a dramatic sodium influx, and it just triggers the opening of all the rest of them, the positive feed forward, the all or none action potential. Over a period of time, each voltage-gated sodium channel is going to inactivate. There's an inactivation gate. Think of a dog shaking from head to tail. When they finally shake their tail, boom, then the inactivation gate closes. So time dependent for closure. Then it's inactive, and it has to continue, readjusting, relax the tail finally, close the dog's mouth and relax the tail, I guess, if you want to think of it that way, after the wet dog shake. Sodium can't come in, even at this point, sodium cannot come in, and it's during this time when they are opened and inactivated that you cannot reopen these no matter how much electrical current you put into them. So it's based on the population of voltage-gated sodium channels at the axon hillock. Have enough of them, like moving back into New Orleans, which the whole city's flooded at this time in 1995. Are there enough houses available for enough people to move in to call it a neighborhood again? So think of it that sense. You've got a whole neighborhood of these houses. Are there any of them that are available to move in and be ready to be activated again? A large enough population to generate this increase in permeability, the tremendous sodium influx to cause the all or none action potential. And the potassium ion channel is voltage gated, voltage dependent for opening, it's time delayed, and voltage sensitive for closing. It can be affected, this voltage gated potassium channel can be affected, or other potassium channels can be affected, so that we can eliminate or reduce the after hyperpolarization, such that the relative refractory period is reduced. This is a figure legend for that. The other thing that can happen, of course, is the absolute refractory period can be changed. Again, I'm showing you the ventricular muscle action potential, and it's this plateau phase that we're considering. So that phase 2, the plateau, the sodium channels are closed, which we know happens at the peak of the action potential. So what keeps the membrane depolarized? They are calcium channels that maintain a depolarizing current, so there's a sufficient calcium influx to sustain that positive membrane potential. It's not the maximum like sodium can, but it's still pretty high. Gradually tapers for two reasons, because the inward or anomalous rectification will turn off the potassium channels instead of turning them on. Therefore, the weak calcium current can keep the membrane depolarized. So once the intracellular calcium levels reach a certain concentration, there'll be a self-negative feedback, an auto-inhibition. They'll close themselves, calcium-dependent closure, voltage-gated, voltage-dependent for opening, calcium-dependent for closing, intracellular calcium concentration. And then these potassium channels are special ones that are calcium-dependent and voltage-dependent for opening. So the voltage change at threshold will get things started, but you've got to have a high enough intracellular calcium level in order to open them. And then this calcium concentration will be reduced through transport mechanisms to remove the calcium, the free calcium, from the cytoplasm, and then these channels become voltage dependent for closing. This slide is showing us the action potential again, the relative refractory period, and we talk about increasing the absolute refractory period, what can happen, or decrease the after hyperpolarization, what can happen, and how can you accomplish decreasing the after hyperpolarization and what can happen. So this set of slides here is showing an action potential. The after hyperpolarization would be positioned right here. So it's the mirror image of this because it is moving down. If you're standing here, you'd be at time 0. You see resting membrane potential. You're right here. And now you're beginning to see the rising phase right here, first part of the absolute refractory period. Now you're here at this time point, and you're at the after hyperpolarization. Right? So this is directional movement, time 0 here, and the other. So this would have to be rotated around. Take this and put it there for the action potential. We got another slide that makes that point as well. I'm just playing with your imagination here. You're standing on an axon. What do you see first? You see the this front end of the action potential. So what is happening along the length of an axon? Of course, the soma would be over here, the axon hillock, and the action potential is moving down the axon in the anterograde fashion. So we have millivolts here and distance on the x-axis. This is millivolts and time. So what we see is at the physical point on the axon where you have the peak action potential, you've got the largest population of voltage-gated sodium channels open. As it is moving away in its trail, the tail end of the action potential, you have closing and closed voltage-gated sodium channels. So you're somewhere in here, you're in the relative refractory period. They're not showing the after hyperpolarization, but this is where it is. Are there, how far away are you from threshold? And are there enough voltage-gated sodium channels available to open to generate an action potential? You might be able to push it and get some of them to open, and that means that you just wouldn't have as great a slope in the rising phase, and you might not reach the same peak. You may only go to 0 rather than plus 30, something like that. So you can have a different shape of the action potential in the absolute refractory period. It just depends upon the population of voltage-gated sodium channels. I know that sounds pretty complex, but if you think about the population, and you have to have a critical number opened to produce the action potential that we classically study like this. So as this continues to move along, we see the relative refractory period here now, and I guess this light blue is all relative refractory period. And then as we're moving away, the portion that just underwent the relative refractory period is now back at resting membrane potential, and you can have another action potential occurring here. So with the after hyperpolarization in the relative refractory period, this is the closest distance you can have between two action potentials. If you were measuring them coming past here, then it would be how many per second to generate the frequency. So it would be under normal conditions with a typical relative refractory period and after hyperpolarization, that would be the maximum firing rate you could get out of a neuron. So what we're thinking is, what if we impact the voltage-gated potassium channels and alter the after hyperpolarization? Can we get another action potential trailing closer to the action potential that just passed this portion of the membrane? So it'll depend on the population of voltage-gated sodium channels being reset, as well as, of course, the membrane potential being closer to threshold. So this is showing two action potentials along the length of an axon. So again, we have membrane voltage on the y-axis, time on the x-axis for these action potentials, and then distance in centimeters on the axon itself. So again, if you were standing here, what you would see first as this comes at you is the rapid rising phase, then the repolarizing phase, the after hyperpolarization. So this is the minimum refractory period here at the after hyperpolarization. We cannot have an action potential any closer than somewhere in here because we have to have the membrane back to resting membrane potential or very close to it, and a large enough number of voltage-gated sodium channels reset so that they can be activated and we can have another action potential. So we can see here that we have basically 10 milliseconds and two action potentials. So what would be the firing rate of that neuron? So we need millisecond is 1,000th of a second. So we're getting 10, can that be right? 10,000 action potentials per second, 10 kilohertz. That's pretty fast for a neuron. I think something's going on here. Anyway, let's just not worry about that for now. But what we're seeing is that the after hyperpolarization is something you can target to impact the highest frequency of a pulse of action potentials leaving a neuron at any one point in time. Now let's think of neurotransmitter deactivation. After the neurotransmitter stimulates a postsynaptic receptor, it will, it's a dynamic binding, binds the receptor, unbinds the receptor, so it is basically, if we are at a balanced point, it's available in the synaptic interstitial fluid as often as it is in the bound form, and if it's available in the synaptic interstitial fluid, it can be acted on by something else. So it can either be deactivated by removal, so reuptake. So there's so 80% of the catecholamines are removed from the synaptic terminal through the reuptake proteins on the presynaptic axon terminal. So there is a dopamine transporter, DAT, on the presynaptic membrane of dopamine neurons. There's a noradrenergic transport protein on the presynaptic membrane of noradrenergic neurons, so NAT. There is a serotonin transport protein for reuptake of serotonin at a serotonergic synapse. So it's the acronym for serotonin reuptake protein is SERT, SERT. So those are prime examples. And the catabolizing enzymes are also available at different locations, either within the synapse, as is the case of the acetylcholinesterase and the catechol-O-methyltransferase, or within the presynaptic or postsynaptic neurons, such as with the monoamine oxidase, and GABA transaminase, I believe, is in the presynaptic terminal as well. So there are many dynamic processes that contribute to synaptic concentration of a transmitter, and of course it's the synaptic concentration that determines how frequently any transmitter molecule is likely to collide with and bind to its receptor or reuptake protein or catabolic enzyme. Remember, they're all just in there bouncing around, so you're either going to hit your postsynaptic receptor or presynaptic reuptake protein, or bounce into an enzyme that's available in the synapse. Actually, the acetylcholinesterase is embedded in the postsynaptic membrane. I should clarify that. So anyway, let's not get too burdened by where these are all located. We'll make a few points as we proceed. So this figure from the text is showing the degradation of noradrenaline from a noradrenergic neuron. So we know that the vesicles, synaptic vesicles, contain both dopamine beta hydroxylase and noradrenaline. Both of these are released with exocytosis into the synapse. It is known that dopamine beta hydroxylase is taken back up with the presynaptic membrane to regenerate vesicles that would have the VMAT on them as well, and would be capable of being recharged with dopamine and converting the dopamine to norepinephrine to contribute to the releasable pool of vesicles. While the norepinephrine are showing it being taken back up and transported into the vesicles here, while the dopamine, while the norepinephrine is in the axoplasm here, it is available for, before the VMAT can act on it to put the noradrenaline directly back into and recycle it back into the vesicle for re-release. It is available to collide with, again, dependent upon concentration and diffusion around in here. If it bumps into the monoamine oxidase on the mitochondria, it will be degraded. And the different metabolites are produced by the different neurotransmitters. We won't be concerned at this moment with those different metabolites, but some of you may know them. The catabolite for the catecholamines, whether it's converted by monoamine oxidase, catabolized by monoamine oxidase, or the catechol-O-methyltransferase, will be different. There's similarly, the monoamine oxidase will degrade serotonin into 5-HTT, 5-hydroxy. Oh, I don't remember that one. So forgive me. Okay, so we see all the events bind to the postsynaptic receptor, but again, they don't have determination. They don't decide where they're going to go. They're diffusing down their concentration gradient, higher concentration in the vesicle versus the synapse. They're out here, they're just diffusing around, bumping into everything. They might bump into the presynaptic membrane and be taken back up. In here, they might bump into the membrane and VMAT will recharge a vesicle, or they might bump into monoamine oxidase and be catabolized. Might also bounce around, bump into a presynaptic receptor. There are autoinhibitors. Might bump into the postsynaptic receptor. So synaptic concentration is extremely important. Low synaptic concentration means you're going to have a rarity or low frequency of any and all of these events. Greater release or impact through a variety of mechanisms on this that increase synaptic concentration are going to increase all of these events depending upon what you're doing. You may be blocking the NAT, noradrenergic transporter, as with some of the classical antidepressant molecules. The tricyclic antidepressants will inhibit the noradrenaline as well as the other monoamine reuptake transport proteins. And there is a table in the textbook that I haven't included in this lecture slide set that you should familiarize yourself with. I will present this simple version of how to think about it, and then you can work on memorizing from the table the different, and there's a figure as well, the different methods of increasing or decreasing events at the synapse. I focus here on changing synaptic concentration of the neurotransmitters. So you have input and removal. It's like putting water in a bucket. You're dumping water in from above. You've got to have a source to contribute. So you've got to synthesize. You can increase synthesis. You've got to have an increased storage amount. So the vesicular transport is important. You can increase release, dump more from your source, and this can be brought about by blocking those autocrine receptors, those autoinhibitory receptors on the presynaptic membrane, the presynaptic autoinhibitory receptors that will increase release. It's one example of how you can increase release. You can increase the firing rate of the neuron as well, as another example. So this will contribute on the input side. So then you've got, remember from the synapse too, it can diffuse away, but you've got the removal side, decrease reuptake. If you block the reuptake, like we just talked about with the noradrenaline transport molecule, or the SSRIs for serotonin, selective serotonin reuptake inhibitors. Many of the antidepressants, the newer antidepressants, target just the SERT, serotonin reuptake protein, transport protein. So you decrease reuptake, you stay in the synapse longer. So you've got a hole in the bucket you're dumping into, so that's the removal side. Or a straw that's sucking water out, I guess, the two type. Straw sucking it out is catabolism, and the reuptake, you know, the hole in the bottom bucket being piped back up to your source bucket. I don't know. Work out the model there. If you decrease reuptake, you're obviously going to get a buildup of transmitter in the synapse. If you can target the catabolic enzymes, that also would do it. Remember, these are all dynamic processes. If you just do this, then the catabolic enzymes are going to have a higher concentration, greater likelihood of collision, binding to the biologically active site of the catabolic enzymes, and the degradation rate will increase as well, but you're still going to have an increased synaptic concentration. Now I'll just end this discussion by pointing out a few things that I'd like you to familiarize yourself with the various neurotransmitter pathways. First, here we see the cholinergic pathway in this sagittal section of the rat forebrain, the olfactory bulb at the rostral end at the nose, and the medulla at the junction with the spinal cord. You can see that there are these basal subcortical cholinergic neurons, so it's all the cerebral cortex up here. There's the hippocampus. So this basal nuclear complex is the one that is targeted heavily in Alzheimer's disease. Projections to the cortex, to the limbic area, and to the hippocampus, so traumatic impacts on memory function there, of course. And then there is this brainstem, the mesopontine, and the midbrain pontine roof tegmentum complex that is very important for behaviors. These descending cholinergic neurons contribute to innervate neurons in the spinal cord and contribute to coordinated motor behaviors. Obviously, there has to be an extensive cholinergic innervation of the hypothalamus. We included discussion of acetylcholine having effects on many different hypothalamic hormones. For example, the oxytocin, so sensory information from the nipples, or that's a somatic sensory information, or visceral from the uterus, the cervix specifically, ascend the spinal cord and somewhere impinge upon cholinergic neurons. I don't know if it's actually here, and those cholinergic neurons would dump acetylcholine onto the oxytocin neurons, increasing the firing rate and increasing the release of oxytocin at the posterior pituitary, and so on and so forth. Here are the dopaminergic pathways, and I want you to recognize this nomenclature of the A. It originated, I guess, as aminergic, monoaminergic, so they're all three catecholamine systems are labeled A. So there's A8, A9, and A10. The A10 is the ventral tegmental area, and that innervates, provides a lot of the dopamine to the hypothalamus, but also to the reward areas such as the nucleus accumbens that we had mentioned, as well to the hippocampus and some of the cortical areas. The substantia nigra is the A9 collection of neurons innervating the, providing the nigrostriatal pathway to the subcortical motor areas such as the striatum, nigrostriatal. And then there's the tubero-infundibular pathways that we've talked about for dopamine release at the median eminence to affect the anterior pituitary. The noradrenergic pathways are shown here. So there is the A1, A2, A4, A5, A6, and A7. The locus ceruleus is the A6, and that's the one that we've talked about, bringing noradrenergic innervation to many of the hypothalamic neurons, but also the A1 and the A2 noradrenergic populations of the medulla oblongata. For example, the A2 in the nucleus of the solitary tract, or the nucleus tractus solitarius, to put it in Latin for the acronym NTS, also bring a lot of visceral sensory information up to the neuroendocrine hypothalamus, and as well to many of the cortical areas, including the hippocampus. So we're coordinating cognition, behavior, autonomic nervous system, and neuroendocrine functions through these pathways. Keep those in mind. The serotonergic system, not sure the origin of the B for this, biogenic amine, I guess, I don't know. There's a B1, B2, B3, B4, B4 what? Anyway, 5, 6, 7, 8, and 9. So the RAF-A nuclei include the B3, 4, 5, 6, and looks like the 9 that they're including there. So it is a relatively small population of serotonergic neurons. These are the dorsal RAF-A neurons here above, the B4 in particular. And many of these serotonergic neuron populations are the source of the innervation to the hypothalamus. As well, they go to the motor areas, reward areas, and cognitive areas. So all these monoamines providing neurotransmitter as well as, and more often, neuromodulatory effects. Again, keep in mind my definition of neuromodulation is more along the lines of metabotropic receptor-mediated changes in target neurons that may be synaptic or extrasynaptic. So my definition differs just a bit from the textbook. But if I ask you a question about the distinction between neurotransmitter and neuromodulator, use the textbook, strict definitions, non-synaptic for modulator. And then here we're just seeing the coronal section, midline section of the human brain. Obviously looks different than the rat, much more cortex. But the dopamine neurons are in the same location, the substantia nigra, or the A9 and the A10 of the midbrain tegmentum, or the ventral tegmentum as it's called. You go in this direction, it's called ventral. Go that direction, it's called dorsal, rostral, and caudal, toward the nose and toward the tail. So ventral mesencephalic, ventral tegmental area, ventral tegmental area, the A10 reward dopamine neurons going to the nucleus accumbens, prefrontal cortex, and then of course the nigrostriatal are shown there. So the text, as I said, provides you a discussion of the effect of drugs on neurotransmitters. So neuromodulators can alter deactivating enzymes like monoamine oxidase or catechol-O-methyltransferase. It can mimic or block the neurotransmitter as agonist or antagonist. It can be a false neurotransmitter. It can bind to postsynaptic receptors as an agonist or an antagonist, or presynaptic receptors. So there's a wide variety of potential effects. You should familiarize yourself with those and feel confident in understanding them for the style of questions that are asked in this class. Either fill in the blank or multiple choice and matching. So there can be effects of nutrients such as carbohydrates, elevating tryptophan, driving serotonin synthesis, and impacting sleep, mood, and appetite. High protein levels increase choline and contribute to attention and memory effects. Foods high in tyrosine can impact dopamine and other catecholamines, cause a reduction of uncontrolled movement in Parkinson's disease. These are quite far-reaching. They're not to be ignored. Definitely, if tryptophan is not present in the diet, or if tyrosine is not present in the diet, if there's a severe deficiency, then there is a decrease in the synthesis of the corresponding neurotransmitters. So I'm not belittling this or minimizing this completely, but you're not going to cure diseases by diet in this approach that's implicated here. And then finally, the divisions of the nervous system. There's the central nervous system. I'm assuming that you come into this course with a strong enough background to review this material for yourself, understanding the nerve cells and the neural pathways that we're implicating in our discussions. Peripheral nervous system, I do want to talk about a little bit, the sensory and motor. There's the somatic or the visceral. So the somatic is the voluntary, the visceral is the involuntary, mediated through the autonomic nervous system. So both receive sensory input, but from different organs, musculoskeletal, and the skin, mostly from the somatic, as you know. The different organs, intestines, heart, arteries, gastrointestinal system indicated there. So there's visceral sensory and somatic sensory information. We differentiated for control of oxytocin release, mechanoreceptors in the nipple would be somatic sensory input. From the stretching cervix would be visceral sensory input, converging ultimately on the hypothalamus, mediated in part at least through a common neurotransmitter, the acetylcholine. So we try to put this together as best as we can. The hypothalamus has descending projections that impact other collections of neurons along the way through the brainstem and extend all the way down to the spinal cord to impact the autonomic nervous system and can either increase or decrease sympathetic or parasympathetic nerve activity through the systems that we know and understand and continue to develop through this course and our other studies. There's also a separate enteric nervous system that's really quite complex, We know it's made up of sympathetic and parasympathetic components, but it is really a very sophisticated system and can almost be considered as a separate one all in and of itself. So there is this illustration of the parasympathetic and the sympathetic nervous system. The parasympathetic output neurons, or efferent neurons, preganglionic because they synapse on a peripheral neuron and a ganglion, are in the cranial motor nerve nuclei, visceral motor nerve nuclei, and S234, so it's the craniosacral system. And the sympathetic neurons, the final output neuron, final common pathway, is in the thoracolumbar region, so it's called the thoracolumbar system, coming from the spinal cord only. And also these are preganglionic neurons because they target neurons in ganglia in different locations, the postganglionic sympathetic neurons. There is the exception of the direct innervation. I'm trying to find it. Goes straight to the adrenal medulla, and that's one that we'll consider with acetylcholine being released by all of these sympathetic and parasympathetic preganglionic neurons, and acetylcholine being released by postganglionic parasympathetic neurons, synthesized and released, whereas noradrenaline, norepinephrine, is synthesized and released by the postganglionic sympathetic neurons. So I challenge you to refresh your memory on these aspects of the autonomic nervous system, and we'll try to discuss them as often as we can. Certainly the acetylcholinergic innervation of the adrenal medulla targeting nicotinic cholinergic receptors, and we'll have a separate discussion of that when we talk about receptors.